Welcome to week 11 of BIOT 643. This will be our last lecture. In this lecture, we'll be discussing synthetic biology. The topics we'll be covering this week are an overview of synthetic biology, abstraction, synthetic biology open language, the rational design for gene expression systems, molecular logic circuits, and applications. So first, what is synthetic biology? Synthetic biology is the design and construction of new biological parts, devices, and systems. It can also be defined as the redesign of existing natural biologic systems for a useful purpose. Overall, it is the combination of engineering with biology to engineer living things to create novel applications. These can include fuels, medicines, and other materials. Synthetic biology is an emerging field of biology that aims at designing and building novel biologic systems. The final goal is to be able to design biological systems in the same way engineers design electronic or mechanical systems. It is one of the most exciting areas at the forefront of biotechnology. It is an interdisciplinary area that overlaps the areas of biophysics, computer engineering, molecular biology, synthetic chemistry, computational biology, microbiology, programming, and systems biology. The main goals of synthetic biology are to apply rational design to construct new biologic systems that are not found in nature, or to redesign existing natural biologic systems for useful purposes. Synthetic biology treats biologic systems like other engineering systems by reducing to parts that which can be combined into larger systems. For example, like computer programs with the codes, subroutines, complex programs. In this way, they compare computers to cells. By applying the principles of engineering, we can achieve the standardization of parts, designs, and circuits that are necessary to develop biology into a true engineering discipline. This is achieved by comparing the engineering of electrical systems to the engineering of a biological system. One of the key requirements is standardization and interchangeable parts. If a machine requires a bolt, it really doesn't matter where you buy the bolt from. A specific size bolt should be exactly the same regardless of the manufacturer. Although biological parts are inherently more difficult to standardize, significant strides have been made. Standardized biological parts would meet design specifications and behave reproducibly from lab to lab. Here on the right, we show an example of this kind of standardization that is currently being developed. The ideal for synthetic biology is to treat genetic features like Lego bricks that can be easily attached and reassembled into different combinations, such as promoters, ribosome binding sites, repressors, fluorescent proteins, enhancers, terminators, and the like. These same components can be assembled and reassembled in different combinations, such as to say protein generator or a protein fusion generator or a monocytronic protein generator. The ultimate goal is to have a kit with build your own cell with synthetic biology 2.0, the next generation of bioengineering. So for some examples of synthetic biology, at the International Genetic Engineering Machine Competition in 2012, here were some of the winning competitions. There was a system called the E-Chromi, which is an E. coli that has been genetically modified for toxin detection and variable level reporting. This system was based on the E. coli having various chromophores added depending upon the toxin detected. The amount and intensity of the chromophore expressed gave you a color readout, as shown here. In addition, there were genetically engineered common fungi that produced biodiesel fuel from cellulose waste. There was an E. colonoscopy system that used genetically modified E. coli as an early detection system for colon cancer. They developed the E. coli system with a red fluorescent nitrate detector and a bacterial dosimeter, enabling the detection of harmful radiation. The next topic is abstraction. The goal for synthetic biology abstraction is to simplify the engineering of complex systems by ignoring unnecessary details. Why is this necessary? Well, for simplicity and ease of handling. Here, for example, we show the sequence of the LAC-Z gene. This is unnecessarily detailed. But using this symbol is much simpler and ultimately much better. Abstraction allows for easy conceptual understanding. It makes it easier to edit and rearrange devices and systems. It's far less cumbersome than dealing directly with the actual DNA sequence. Here, we show a pathway in which we incorporate the gene for GFP. So in this manner, we can show how multiple genes can be transduced or placed into the gap left by the GFP. Here we show a few examples of some of the symbols that are used for abstraction, such as for the ribosomal binding sites, regulatory pathways, RNA, DNA, protein coding, terminators. Synthetic biology abstraction hierarchy. There are 4 increasingly complex levels. 1 is DNA. It is the most basic unit, the actual nucleotide sequence, such as shown here. This leads to a part. This could be a collection of DNA nucleotides, i.e. a sequence, that encodes a specific feature of biologic significance. It's represented by a symbol that indicates its function, such as a coding sequence, an inducible promoter, a terminator, or a ribosome binding site. We then have a device. This is a collection of parts that together perform a specific function. Here we show the entire pathway of synthesizing the GFP protein. You have the promoter, the initiation step, the promoter, the ribosomal binding site, and the termination site, which leads to the production of the fluorescent green protein. And last is the system. This is a collection of devices that together perform complex interrelated functions. And here we show a hypothetical system where we have multiple genes being expressed together in a pathway, such that this would be device 1 that produces a protein that binds a toxin in the environment, for example. This protein undergoes a conformational change when it binds the toxin. That brings us to device 2, where the bound protein toxin complex binds to the promoter upstream of the GFP. This now leads to the expression of the fluorescent green GFP, which leads to device 3. The bound protein also binds to the promoter upstream of a gene that inactivates the toxin. So the design system detects the toxin and results in 2 responses. First, a visual signal that allows or defines the presence of the toxin, and a second signal that acts to inactivate the toxin. So here we could show that in a flow diagram. So here we show the possible hierarchy for synthetic biology. One of the important things to remember is that the goal of this is to compare classical engineering pathways to biologic ones. In this case, we show a computer network as being similar to tissues, where you have the parts, that the physical layer that leads to gates that are combined to make modules that are combined to make computers. These computers are interfaced together to form networks. The same way you have proteins and genes combined together to create biochemical reactions. These biochemical reactions interact to form pathways. The working together and the interaction of these pathways are essential for the survival and production of cells, and the cells interact together to form the tissues. So the same way we just talked about there being parts, here we show the parts. For in this example, the promoter, the operator, the structural genes, the regulatory genes are all parts of a device. Here we show the systems or modules where we have all of these individual parts coming together to form an activity. And one of the ways that we describe how these networks fit together is synthetic biology open language. In order to ensure that synthetic biology designs are compatible across thousands of researchers using many different software programs, everyone needs to be speaking the same language. To that end, Synthetic biology open language has been developed and curated by some of the top names in the synthetic biology community. This enables a data exchange standard for descriptions of genetic parts, devices, and systems, as well as visualization and the development of software tools. One of those softwares is Tinkercell, which allows you to create pathways such as shown here. The synthetic biology open language visual is a graphical component. It uses specific symbols to represent abstracted genetic parts. Here we show a few examples, such as that used for a promoter, or the origin of replication, for an operator, for a terminator, for ribosome entry site, for blunt restriction sites, for sticky restriction sites. The synthetic biology open language and the open language visual have undergone periodic revisions to maximize their usefulness. The Open Language 2.0.0 was released in July 31st of 2015. Most notably, the latest version now supports modeling of interactions between various molecules. And here, on the right diagram, we show how the new version has extended some of the ways of visualizing these metabolic pathways. So, rational design of expression systems. What is rational design? If you want to design a specific function, there are two main methods. One, you can make changes to naturally existing complex systems, likely with unpredictability and possible unintended consequences. This is a top-down design, and this shows you a traditional metabolic pathway. Or two, you can use rational design, the concept of starting with a desired system and building it piece by piece using specific well-characterized parts. This is what we call a bottom-up design. And by piecing together each of these individual parts, we can now complete an entire device as well as system. The advantages of rational design are that it can specifically tailor genetic design to desired outcomes. Positively induced promoters versus negatively induced promoters, strong promoters versus weak promoters, strong ribosomal binding sites versus weak ribosomal binding sites. There is a greater predictability through the use of characterized parts. You can avoid upsetting the delicate balance of existing systems, as these design systems can be designed to be orthologous to the existing host genetic background. There are some disadvantages. It is limited by the number of available parts. Used parts should not interfere with each other, i.e., inducing the same chemical. So, orthologous genetic circuits. These pathways are one which have no effect on the biology of the existing host and are unaffected by the biology of the existing host. Because of this, they can be introduced into a heterologous system and maintain a complete independence from the existing host biology. For example, the RNA polymerase from the T7 bacteriophage. So the T7 is a highly efficient promoter from the T7 bacteriophage. It is not recognized by most RNA polymerases other than the RNA polymerase encoded by the T7 phage itself. Additionally, the T7 RNA polymerase recognizes no promoters except the T7. Because of these specificities, the T7 and the T7 RNA polymerase system is referred to as an orthologous system, meaning it can be expressed in an organism without affecting the organism's native regulatory system or being affected by those same systems. This makes the T7 RNA polymerase system one of the most useful and frequently utilized in molecular biology. One excellent source of standardized biologic parts is the Registry of Standard Biologic Parts, which is founded at and initially operated by MIT. It now operates as an independent nonprofit entity. And this is the kind of information that the registry provides you on the individual parts and DNA collections that it provides. Again, at the bottom shows the website in case you're interested in visiting that site. So now we'll talk about molecular logic circuits. Logic gates are used in electrical and computer engineering to perform logical comparisons and calculations. For an example, there is one that is called an AND gate, a logic gate that has two inputs and a single output. Here in this diagram at the bottom, we show you this AND gate. When both inputs equal 0, that means off, the processed output of an AND gate also equals 0. In the case where either input equals 0 and the other equals 1, that is on, the output still is 0. Only when both inputs are equal to 1 is the output of the AND gate equal to 1. That is when both input A and input B equal 1, hence the name. There are various logic gates that exist, which interpret input data differently and result in different outputs. And here in this figure we show a whole series of these, whether it's or and, or not. Even more complex calculations can be performed when the logic gates are connected in combinations. And again, here we show an example of an AND gate connected with two OR gates that lead to an AND or a NOT gate. By connecting promoter systems and their products, similar molecular logic gates can be created. For example, a molecular AND gate that produces GFP only when both salicylate and arabinose are present in the environment. Here we show such a system where you use a promoter that recognizes salicylate as well as a promoter which binds the arabinose. Both promoters must be activated in order to result in the production of the GFP protein. So what are the applications of synthetic biology? Synthetic biology has grown by leaps and bounds in just the last few years. It has nearly unlimited potential. Just a few of the many potentials are bio-based chemicals, biofuels, bioremediation, biosensors, industrial enzymes, information storage, improved food nutrition, improved food yields, molecular medical devices, novel antimicrobials, pharmaceuticals, vaccines, and antibody productions, as well as many more. So here is an example: malaria and antimalarial drugs. Malaria is a disease caused by numerous parasitic species, and the parasites are introduced into humans through a bite of mosquitoes from the genus Anopheles. In 2013, 198 million cases of malaria were seen worldwide, with 500,000 deaths, mostly in children in the sub-Saharan Africa. Artemisinin is one of the most potential anti-malarial drugs. It's highly effective, it is well tolerated with few side effects. It is the natural source of artemisinin is the sweet wormwood plant, but the worldwide supplies of this plant are insufficient to meet the demand. It is grown mostly in China, and processing to extract the artemisinin can take up to 18 months, and it is subject to extreme price fluctuations. Even in 2007, we saw a price increase from $200 to $1,100 per kilogram. Perhaps the best known success to date for synthetic biology is the production of artemisinin. In the laboratory at the University of California, Berkeley, they successfully inserted genes into yeast cells, allowing synthetic production of arsenic acid, the precursor to artemisinin. This production method, enabled by synthetic biology, is expected to supply around 1/3 of the global need for artemisinin, while simultaneously decreasing the per dose cost significantly. Biology offers a range of molecules that can be manipulated to produce useful materials or devices. Synthetic biology incorporates engineering approaches to take advantage of and improve biologic systems to tackle significant problems. Protein engineering manipulates protein production, incorporating modifications to improve proteins. Recombinant proteins can provide much information about protein function, both in vitro and in vivo. Engineered proteins have huge potentials in biotechnology and medicine.